The RAS gene is going to be coding for G proteins that are going to play an important role in the signal transduction of RAS-MAPK pathway. Mutations in the RAS gene are commonly found in cancers, most commonly in colorectal and pancreatic cancers. The way RAS protein is going to be activated is going to be through the binding of growth factor ligand into the tyrosine kinase receptor found on the cell membrane. This is going to lead to an autophosphorylation that is going to lead to a binding of a protein complex. This is then going to favor the removal of GDP and is going to favor the binding of GTP. Now, the RAS protein is going to be found in two states. One is going to be the inactive GDP state, and the other is going to be the active GTP state. Once RAS protein is in the active GTP state, it is going to lead to a phosphorylation cascade, which is ultimately going to produce MAP kinase. MAP kinase is then going into the nucleus, which is then going to influence transcription factors, which is then going to lead to gene activation. Now, this leads to cell proliferation, and so because it leads to cell proliferation, we need a way to regulate this cascade so that we don't get cancer. And the way this is done is going to be through the enzyme RAS-GDPase. RAS-GDPase is going to convert active GDP into inactive GDP. If there is a mutation in that enzyme, then we're going to have high levels of active GTP. So as a result, we can end up with cell proliferation without hormone signaling, which can ultimately lead to cancer.